Pat Edwards has been at the LMB for just over 40 years, joining the lab in November 1981 as a research technician studying muscle structure with Murray Stewart and Hugh Huxley. From 1985 to 1995, she worked on a project studying Alzheimer's disease with Claude Wischik, and then moved to Gebhard Schertler's lab, where she remained until 2009. In her time at the LMB, Pat has worked on numerous cutting-edge research projects. Her skills at picking crystals of rhodopsin assisted Gebhard Schertler in determining one of the first structures of a G-protein coupled receptor, and she also picked the crystals for the first structure of the beta-1 adrenergic receptor. Pat is now a research support and lab manager in Chris Tate's group, and is also an active member of the LMB Green Committee, who are dedicated to finding and implementing new ways to make the lab more sustainable. Welcome, Pat.
Thank you.
I'd like to start off by going back to the very beginning of your time at the LMB. So you came to the lab after an applied biology degree at Thames Poly, and this was your first job.
Yes, I did a sandwich course degree, so I had a year out at Shell. Actually had labs down in Kent, so that was a sort of job, but it was really studying. But this was my first real job. I made a few applications, and I got an interview with Murray.
What were your very early impressions of the lab then? Can you remember coming for interview?
I certainly can, yes, I can remember. Back then it was hard to find out much information about places. I think now everything's on the web and you can find out loads of information. But I came for the interview. Murray was very nice to me, and we walked around the labs. And back in the old building, he was based on the ground floor near stores and near the X-ray sets. And I remember being shown around, and he actually took me into the X-ray sets and started talking about Max Perutz and hemoglobin. And I remember thinking, oh, I think I'm somewhere important. There's something important that's been going on here. It was quite an eye-opener, actually.
So you had no idea of anything about the place, really?
I had a little idea, I think, but nothing quite as important as that. There's one thing that did strike me at the time.
Yeah. So how was it coming to Cambridge from London then?
It felt like a backwater, to be honest, because Cambridge was in a way much smaller then. I think much more student-based. So if you weren't a student and part of the student scene, I think Cambridge was small. I'd come from South East London, which is still a suburb, but it seemed like there was so much more going on. I remember not having much to do, not much social things to do. I remember having to go to a phone box to phone my parents to say I was all right or something, or my boyfriend at the time. But it just seemed I was on the north side of Cambridge, and it just out of town. It just seemed a bit of a backwater.
So were you worried that you weren't going to like it to begin with?
I think my main memory is probably of just getting used to the place. I think I was surprised by it. I suppose it has such a big name. I'd never been to Cambridge before the interview, so I didn't know much more about it. I don't remember being worried. I think the people around me basically were nice, and I enjoyed it. I certainly remember enjoying it from the off. People were nice to me. So,
were there other people of your age here who weren't students?
Oh, of my age, there were later, certainly, that I remember. My other peers were probably slightly older than me, and by that I mean technicians. Most of the scientists, I guess Murray was probably in his 30s at that point. I don't actually know, I can't remember. So there were people who were slightly older than me, perhaps not my age, because I was only 22 actually, which seems surprisingly young now, really. But and it took a while, to get to know people, I think, because it just does. It always does. I think everywhere it takes more than a year probably to get to know somewhere or find friends. But people in the other labs were very friendly, and I think I felt fairly at home quite quickly.
So who were the big personalities in the LMB in those days then?
Well, Hugh Huxley, I suppose, was fairly famous. Well, he was famous, not fairly famous. But he didn't talk a lot. So, no, he wasn't a very chatty guy. So I had projects with Hugh, but didn't have... When I talked to him, it was very much about work. He kept himself... I can't remember if he was head of structural studies at that time, but quite aloof is what I remember of him. Within our small community, in a muscle group, I suppose you could say, there was Alan Weeds and John Kendrick Jones. They were the other group leaders. We were sort of the muscle group, Murray, and the other two groups. Max was obviously always there, but from afar, I think people were in the canteen. So Max would have been there. He's probably the main one I would remember at that time.
So did you socialise with structural studies people, or was it everybody?
Mostly, I would say structural studies probably, and certainly initially. People would congregate in the canteen and you'd sit at certain tables, usually with your groups, but often in structural studies groups perhaps, or something like that. And it wasn't until... It probably took, actually thinking about it, it probably took me a bit more time to settle down, because I had a boyfriend at the time who was in London, and I used to go home a lot at weekends to see him. So I think the actual socialising part probably came much later. But there were, yes, as I say, there were many people around me that were always friendly, and it was always good fun to be in the labs. I remember it being good chat, entertaining, you know, making jokes, conversation generally, not necessarily about science, but...
So the fabled canteen culture then, where everybody was supposed to be having intense conversations about their work, wasn't just that, presumably?
No, very much. I think it would be, you know, the guys were often what football games they'd been playing at the weekend, very much so. What football results were, what people had maybe watched on the television, things like that, I think. Yes, it was strange actually in the canteen because people smoked as well. There was a lot of, you know, everyone smoked in the canteen, which was fairly awful at the time.
Did people smoke in the labs then?
Yes, they did, yeah. So... when I moved to work with Claude Wischik, he was he smoked French cigarettes, Gitan or something, and he wouldn't smoke much during the day, but he would work all night, and I'd come in and clear away fag ends, Gitan or whatever from the lab. Amazing, and people smoked. You know, I imagine many less people probably smoked as a scientist than might have done in the public, but people did smoke.
I'm always amazed there weren't more fires, actually.
Well, Mark Bretcher famously smoked quite, you know, much later than a lot of other people. But no, there were no incidents, I think, involving cigarettes ever, as far as I'm aware. But, yeah. And the labs were crowded. I remember when walking with Claude, the lab was crowded and piled high with paper.
Were you part of the always here culture as a technician?
No, not so much. I mean, you tended to work relatively longish days, I suppose, but never very long. I've never done the all-nighters. There has been occasional times when I've had to be here in the evening or all night, but I never felt that I had to do that. And as a technician, I think it was a different onus as well, certainly then, because we came in as technicians. The people that were technicians at the time had degrees. We didn't have PhDs. Some people I knew that came in with degrees later then got PhDs. They did part-time degrees. Several people in structural studies that I know went on to do PhDs, and they were allowed to, I think, on a part-time basis, which was very good for them. They wanted to do that, pursue that.
And that didn't appeal to you?
No, I've always been a person who enjoyed doing the science. I'm not the best person to... I prefer to do many things practically than study what... I'm not a very person who's very good at concentrating on one thing, and I think to do a PhD would have required a level of study I just wouldn't have... It would have been hard for me, and I think it would have been painful, because I was always a person who was better at learning as I went on, taking in the information around me, Relatively good memory, I'd say, and then more intuitively maybe putting that experience to work and listening to my peers as well. So a PhD would have been, to be honest, torture for me. The idea of having to write a thesis, really, not so much probably the practical work, the idea of writing a thesis would have been a torture for me.
Yeah,
really.
Yeah. You must be pretty good at the bench.
Relatively. I suppose there are many things you take for granted now when you're teaching people and explaining things. There are many things you just do by nature now. It becomes second nature. Knowing how to deal with instruments or working out what might be wrong. Certainly the things I'm used to, yes, it is second nature.
Do you think there's the career structure for technicians is adequate?
I haven't been much involved in the recent technician commitment things. I'm sort of leaving it to younger people, I think. But I think there has always been a problem in progression, I think, for technicians. I've actually been treated relatively well in terms of pay, I think. But there have been difficult times when you couldn't move on because there wasn't the money or the progression, and then you'd have to re-interview for a job or something of a new job scale. Initially, when I came in, there were there were levels. You worked up through the scales, and that's how it worked. But that changed. I can't remember when. Various civil service. I mean, we were often aligned to the civil service. I don't think that's true anymore. We have our own pay bands now, and so there have been a lot of issues people have come to, and pay obviously at the MRC is much more restrictive than other places. So I think many people have found it difficult to move on.
It seems to me it's such an important part of a lab, having somebody like you around who's got who holds the lab memory and can teach all the new people what to do. There should be a lot more recognition given of the people who do that.
Yes, and I think it might depend who you work with, who your boss is, who your PI is. And I felt that I've always been appreciated. I must admit, I've always felt that I've been appreciated by the people I've worked with, which might mean why I've been here this long, actually, part of it. But it's true, it's about, it's just about passing on some of the boring stuff in the lab that you just take for granted, that people just don't understand, and all those things that you have to show and go ahead with. The things that you do every day that someone new, I think, is important and gets people off and running, because it can be quite imposing and new here.
Yeah, indeed. Do you find yourself acting as a sort of, I don't know, I hesitate to say a maternal figure in the lab, but you know what I mean. Do you think people come to you for emotional support as well as technical support?
They have done. I've often said that Chris, my boss, is like the father and I'm the mother or something, and so I'll often have the more personal conversations, perhaps, or the problems that people are finding, possibly about even the work or something. So I often may have those conversations, it's true. It's different. I mean, my current boss, Chris, he's actually very easy to chat to, so I will always recommend people to talk to him. And he is easy to talk to, but I think the difficulty is sometimes when you see your boss across the other side of the office, you don't want to interrupt him or something, but actually he's very communicative, but there are certain things that you perhaps want to talk to about the more personal stuff or the everyday things, problems and things.
Yeah. I just want to back up a bit and go back to when you started working with Claude Wischik. So this was right at the beginning of when people became interested in Alzheimer's as a biological problem. So what was it like?
It was exciting. I mean, Claude came in, he was a medic. I think he hadn't done his final training, but I can't remember what level it is. But he came in, I think, through Aaron Klug, to work on Alzheimer's, and he was the first person, basically. He had the link to the brain bank to get brain samples, human brain samples, and it was down to him to find out what the problem was. I don't think, it was early days, and it was exciting. I mean,
so it wasn't even clear then what was going wrong in the brain?
No, I think there was the initial thoughts about paired helical filaments, PHFs, and so there was the histology based on the disease. Yeah. But it was basically coming down to the actual experiments and actually getting those PHFs out of samples.
What was it like working for Claude then?
He was a very entertaining bloke. I liked him a lot. He was a bit of a rogue entity. I think a lot of science will moan about medics in research because they can be a bit arrogant and bullheaded and do what they want. And he did have that reputation. He annoyed a lot of people, but I got on with him very well. Actually, I still regard him as a friend. If I wanted to go and see him, I'd go and see him. He moved up to Aberdeen, but I liked him. I liked him a lot. He was always very nice with me, and again, relatively relaxed with me. Initially, we worked hard as a pair, just the two of us, and then he got more staff.
So what were you doing to begin with?
I was doing the more routine preparation of the PHFs from the brains. So I would actually go over to the brain bank in the hospital. We would get the material. I'd have to do some of the first preparation of the brain material over at the brain bank, and then I'd bring back samples to spin, and we'd be using sucrose gradients to purify the PHFs. And many of the first EM micrographs Claude took, and then with Ross Jakes, who was in the lab as well at the time, we started looking for antibodies, and then there was more people came to the lab. There was, his name, Michael, I'll say for the moment, who's an immunologist working with Cesar, and we started trying to get antibodies to the PHFs. So literally purifying them and then injecting rabbits. Then the animal house would be injecting rabbits with PHFs.
How was it working with human brain?
It was all right, to be honest. It was just squidgy stuff. It was a bit weird when you thought about it, really. Yeah. But in the end, actually, most of it had been cut out. So you were taking cortex and then just squashing it. You just had to be a bit careful. Some of the, obviously we were in certain rooms to use it in the initial parts, but it was just a squashy material, I suppose is the best way to describe it, mate.
It wasn't like you were thinking you were squashing up people's lives.
No, not really. I suppose you knew that it was for a good reason, and obviously people had donated their brains, and you had to treat it with respect from that point of view. And the point was to use it for research. So
that was a very important time in Alzheimer's research.
It was. I mean, it was. There were a lot of people very excited about it at the time. Cesar Group was involved. John Walker was involved. Aaron was to a certain extent. He was, I suppose, Claude's probably overall supervisor later on, Tony Crowther. It was an important time, and it was. And Michel Godin arrived a bit later. I can't remember the timings, but so it then expanded. But it was interesting, and of course there was a lot of talk about Creutzfeldt-Jakob at one point. So that was an interesting sideline.
You weren't worried you'd been inhaling?
There was. I mean,
aerosol at that point.
There was some worry certainly, but it seemed to be eating burgers or something at the time. That seemed to be the worst part of it. It was all very scary at one point, but it's more about what you were eating, I think, as opposed to working with the brains and the tissue.
Yeah. Huh. So you moved on to the next destination because Claude left? Or?
Yes, so he, Claude, in the while we were working together, gained his psychiatry consultancy or whatever it is. He trained as a psychiatrist. So he ran the brain bank for a while, and then was offered a chair in Aberdeen in the gerontology department, I think, the professor of gerontology, I think it was. So yes, and that would have been about 95 that he was offered that. So he wanted to move. He took some of the group with him. He offered for me to go with him, but Aberdeen seemed too far for me to go. My mother was at that point living in Cambridge as well, and I just felt that I'm an only child, so I had the responsibility to be my mother, and the idea of going to Aberdeen just seemed a step too far, I think. So because I was on secondment from structural studies, I was offered a couple of posts within structural studies, and one to work with Phil Evans and the one to work with Gebhard Schertler. So, and I think I'd done quite a bit of EM, electron microscopy, starting with Hugh and Murray, and then I was doing negative stain and various things with Claude. And many of the... so at the time, Gebhard was doing 2D crystallography, EM 2D crystallography, so it seemed a good sort of a slide into a slightly different technique of 2D.
So this was the first time that you'd actually started doing things like growing crystals and all that?
Yes, it was amazing, really. I had never done anything like that before. We had many, when I was at Murray, we used to try and purify myosin, because that was the big thing at the time, was to try and crystallise myosin, but we never ever managed to purify it. So that was one of the projects back in the muscle group, but we never got anywhere near, I certainly never got anywhere near it. But... Gebhard was the main thing. They had 2D crystals, so we could look at them on EM grids, but the other project was to make 3D crystals of the bovine rhodopsin. And Gebhard had started getting 3D crystals. He had a dark lab that was in the basement. It had to be done under red light and infrared light. The whole prep had to be done under red light.
Because otherwise the structure would have changed.
Yes, because the rhodopsin would have taken up light and changed. So we had
thought about that before, but
yes.
So it all has to be done basically in the dark.
Yes, it was. Well, it was in like a dark room. So we had photographic lights, red lights, and we had torches that we fitted with little. They had these glycerin filters. I used to make them. Used to fit these glycerin filters to the torches, so you could have things for close work. I remember Gebhard showing me how to do all this, and Gebhard was a lovely guy. He was dyslexic and a bit all over the place. I think when he saw that I could competently set up a plate, he was very happy actually in the dark because you're pipetting quite small samples. So we had another guy, Claudio Villa was in structural studies, and he used to do the early parts of the preps. He and Gebhard initially used to go and get cow eyes and cut the retinas out, but then later on they found a company that would actually sell us bovine retinas. So I'd never actually done a retina prep.
There's a company that sells bovine retina.
Yeah, it was basically, I think it's probably still going. I mean, it was fairly big business at the time. I think it was like $1 a retina or something.
It's quite a niche.
Yeah, it was, but there was probably quite a good sideline. An American company. Heaven knows how easy it is to import those things now. But so I never actually did a retina prep, but Claudia would do the initial part and then give me the protein, and then we start putting it on the actors and or the FPLCs at that time, as they were called, and then purifying them further. And I would take samples from the columns, the gel filtration columns, and then set up the crystals under red light. So
did that mean you were stuck in there all day?
A long part of the day, yes. You'd be fine. You could have music on or something, and you'd do certain parts, and it could be quite long days. But it would be like a dark room, so you could leave it. There was a curtain, and you could leave and the curtain, so you don't have to be in there while things were, spinning or something like this. It'd be fine. But the next thing actually that was in a way more difficult was actually looking at the crystals, because you actually had to look at all the plates. So you'd have these 24 well plates and you'd have to look. And Gebhard managed to set up this camera with an infrared camera. So you'd have the microscope, the camera, and then a little television screen above, where you'd look and follow and look for the crystals. And they were pretty reproducible, actually, the crystals. But the trouble is they were never necessarily very good. Actually getting good crystals was difficult, and that came down to X-raying them, basically. So in the end, I ended up starting to do a bit of crystallography as well, which was interesting. It's one of the famous techniques in structural studies, crystallography, and there I was actually doing some of it. And Gebhard was very happy to show me and let me do these things. So I'd have to actually pick the crystals under red light and then freeze them in liquid nitrogen. And once they were frozen, they could then be handled in daylight, things like that. The crystals were very small, 10 or 20 microns wide needles. They could be quite long, up to a couple of hundred microns or something. But they were too small for the in-house X-ray sets. The in-house X-ray sets needed large crystals. So we started going to synchrotrons, and we most commonly went to the one in France, ESRF, down in Grenoble.
Oh, must have been tough.
It was very exciting, actually. Yes, it was. To begin with, it was great. It was all lovely. It was new. We managed to get to Grenoble, get lots of nice food. This is when it was still the French franc, I think, when we first went. And then we'd be on the synchrotron, and you'd get 24-hour stints, and you'd have to use the whole time, just going through crystal after crystal after crystal.
So literally 24 hours of...
Yeah, 20 hours. I'm not sure the last 4 hours were very useful. Yes, it would certainly be 20 hours. Yeah. You could probably doze off and someone would do something while you were dozing, maybe, or something. But they were very exciting, those, the trips. We did more and more. It became a bit more competitive, and we heard that Palczewski in the US had, there were rumours about other groups, Japanese group having crystals, and it became very competitive at one point. And unfortunately, we were scooped in about the 2000s. But we had some data, but not enough to make a good structure. By that time, Jade Lee was in the group as well, helping us with it. And unfortunately, we were scooped, but that was a pity.
Shame.
Yes, it was a shame.
How was it? I mean, that was an awful lot of work.
It was an awful lot of work, and it was hard, actually, I have to say. There was a period when I would say I was a bit fed up, I must admit, because we were sort of having to work, we were going on lots of synchrotron trips. At one point, that year we made trips to the US, to Japan, to get synchrotron time a couple of times. We went to the US, to APS at Chicago. We went to the ESRF several times. It was hard, actually, that year. And I remember being scooped was hard. It was hard.
Did you ever think about doing something else?
I did, actually. About that time, probably after that, I was beginning to question whether I really wanted to do it. I did have a period when I thought, well, is this really what I want to keep doing? I'd had other sideline things where I'd taught aerobics, for instance. I'd learned to teach aerobics, and I'd done a bit of massage therapy, all the things on the side you do at the weekends. And I remember thinking, maybe physiotherapy might be interesting. I didn't get these ideas. And I also thought about possibly going into the admin side of science, and had a couple of interviews, but didn't. I think I was almost offered a job, but I was at a synchrotron. I couldn't take the phone call, I think, but things could have, so I remember. But in the end, things changed, like many things go in waves, and things got better again. It was hard for a while, and I think Gebhard understood it as well. I mean, were very close. You know, as again, it's that that sort of very close relationship you have with your PI sort of thing. But yes, it got better. You know, and I was happy to stay really. But that was the only time, pretty much, that I was looking for other jobs. But there was actually more of a career change as opposed to somewhere else. But,
I guess at that point you would have had a good run at doing something entirely different, so it would have been a good time to...
Yes, and it became routine. It became because we weren't getting it became hard because we weren't really getting the results we wanted. But you know, then we had sabbatical visitors and other people come in, and things changed and it became more interesting, and there were other structures that were done actually on the rhodopsin crystals.
So you managed to get something out of all that?
Yes, there were some good publications, the JMB publications. And also at the time we started working with Brian Kobilka from Stanford. Gebhard was always the sort of person who was very inclusive. And Brian originally came to the lab when I first worked with Gebhard to work on 2D crystals, because he was trying to work on the structure of beta-adrenergic receptor, beta 2. So Gebhard had maintained a relationship with him, and then when Brian started making 3D crystals, he wasn't getting much help back at Stanford because they were tiny and no one had the expertise in tiny crystals. Everyone would just say, go and make them bigger, go and make them 10 times bigger, but they would never be 10 times bigger. It was difficult. We were always working with microcrystals. So I went to work in Brian's lab for a few days, showed him how to freeze some crystals. He came to synchrotrons with Gebhard and I in France. They would give us samples. We would take his samples to the synchrotron. And we saw some of the first X-ray patterns from Brian's crystals at ESRF, actually, so that was exciting as well.
Yeah. What's the most exciting bit of your career, then, do you think? Don't say aerobics, please.
No, that was just different, really. Although that was something I never thought I would achieve either. But I think the crystals with Brian were probably one of the highlights, really, because people had tried to get the structure of beta-adrenergic receptor before, and there'd been some false claims, and it'd been very exciting. Tony in our lab, Tony and Warren had been working on beta 1. Brian was working on beta 2. So we were all, working towards getting a structure of the adrenergic receptor for a pharmacological reason. So when we saw the crystals with Brian, it was very exciting. Yeah. There's a photo of somewhere staring at a screen, I think. It was exciting.
Yeah. Do structural biologists have eureka moments then, or is it more finally seeing a structure and...
To be honest, I would say seeing some of these crystal patterns were almost, not quite, because the actual processing and the downtime to actually get the final thing takes a lot more time and energy and maths.
It's a very slow eureka moment.
Exactly, but the actual making a crystal of a membrane protein was very difficult, and I think so when I joined Gebhard's lab, it was new, and it was very hard to get a purified membrane protein. Was hard, and let alone to get a crystal, and then to get a crystal that diffracted with a regular pattern was a very special thing. And so they were small eureka moments, I'd say. Yeah.
I want to switch entirely and ask you about the Green Committee. If there was one big thing that you could get everyone to change then, right now, what might it be?
Oh, gosh. Probably, in many ways, more communal use of some things, of reagents. Because what happens is when I see people leave and I'm emptying the freezer with there's so many things that people have bought and never used. I think that might be one thing that would help.
Yeah.
In terms of plastic waste, it's hard. And much of it is where it goes. A lot of it is burnt. And I think the idea of now is to try and push it towards recycling it, or it to being turned into furniture or something, you know, being reused. But when I first started, you were throwing everything in the bin, and then at certain points we had to throw everything into a yellow bin because it was clinical waste. If it was found, the MRC could be prosecuted because it's classed as classified, but it probably just had water in a tube or something. But everything that looked like it was clinical had to go in a yellow bag. Yeah. But now they're starting to think about waste routes and where things like plastic waste can go.
So it's moving, but slowly.
It's moving, but very slowly. But there are more people involved, and it's become more of a general topic rather than a few people just trying to recycle a few plastic bags or something. Yeah.
So what are the best and worst things about having been here for 40 years?
The best things are probably the people. I think a lot of people will say that. I've met people, I mean, I had actually travelled as a child. My father was in the army, my mother was Japanese, and we travelled. But actually, it's quite insular. But when you come here, you meet people from everywhere, you know, people from all countries. And you make friends with them. And, you know, you get to visit them and carry on these conversations as well. I don't see everyone all the time, but I've got friends all over the world. The worst thing is you say goodbye to them. Yeah. You know, after so many years, you say goodbye to a lot of people as well. And there have been many people I probably can't remember, to be honest, because I've seen a lot and been very sociable at times, less so now, I'm older, but been very sociable in the lab and had a lot of good fun, but people move on. Mm. And that's probably, yes, that has been tough many of the times, but But I've got friends from, you know, that first week that I've met that I'm still friends with. Several people, actually, that I'm still in touch with. These Cambridge people, really. Yeah, yeah. But then there are friends across the world.
It's funny, you don't really realise when you meet people when you're 22 or 23 that you might still know them when you're 61 or
yes,
so.
Absolutely.
But yes. It's definitely true in science that people are just there for your whole career sometimes.
Absolutely, yes. Yeah. And, you know, it's depending on certain subjects, you know, people, if you're in a research area, you're friends with many people, you come across them in conferences and things, but for me, it's been more about the relationship in the labs, enjoying people's company, hearing about their side of things.
Yeah. So I want to finish by first of all pointing out that you have published with 6 Nobel laureates, which is a pretty amazing statistic. Yes. But that also brings us neatly onto this bottle. So can you tell me about your champagne bottle, please?
So I came in November 1981. And Aaron won his Nobel Prize in October 1982. Mm-hmm. So I would have been here less than a year when Aaron won his Nobel Prize, which was incredible. I mean, the idea that someone I knew had won a Nobel Prize or someone in our building, it fills you with such pride, actually. I think that's also one of the good bits about being here, is the pride you can feel in the building. But yes, I started taking signatures on these bottles, and then I have...
So this is from Aaron's Nobel Prize celebration.
You'll see a lot of these bottles now. People do it, but I think I was the first. Yeah. I think I was one of the first to do it. So Aaron signed that on the day he won the Nobel Prize. Uh-huh. I haven't got everyone, much to my distress, but I never got Max to sign it, actually.
So people were signing that particular bottle on that time that Aaron won the prize, or it's come out every subsequent?
Aaron signed it, and then I think César won in 1984, and I got him to sign it then. Uh-huh. And then I can't remember. Then I think I got Fred to sign it as a back. Retrospect. Yes. And then I got Greg Winter and Venki to sign it, and Richard to sign it recently.
John Walker, I guess.
And John Walker. Yeah. But John Sulston, I only got him to sign it when Richard won. Uh-huh. I asked John, he was very sweet. He gave me a hug and signed my bottle. So there's a couple of retrospective ones from that point of view, but I never got Max to sign it, which is
that's a shame.
shame, but yeah, that's a pity. But obviously never going to happen. But so it comes out every so often.
Yeah, well, we'll have to see when it comes out next.
Yes, it is, absolutely. Yes, it's a great thing to have.
So you just keep it in the lab?
It's generally in the lab. Yes, it's hidden behind my computer under a book.
Oh, you shouldn't have told us that. You're going to have to move it now.
Well, I think the dust and all the paper or work around it probably hides it anyway. Yeah. But yes, it's signed by Erin.
You should try and do a DNA prep on it. You've probably got quite a lot of interesting genomes on there, actually.
Okay. True. I don't know actually what you could do.
Well, it's a fantastic memento, isn't it?
Yes, it's a special thing. I mean, you begin to take it for granted, stupidly.
Well, you do, really.
Yes, absolutely.
Yeah. Thank you, Pat. That's been really interesting. Thank you so much for coming to talk to us.
Well, thank you very much. That was, yes, I'm glad to do it. Very happy to.